Welcome back. In Talk 1, we covered p-values and confidence intervals, the fundamental language of statistics. In this talk, we're zooming in on drug trials, specifically randomized controlled trials, or RCTs, which are the gold standard for evaluating whether a drug works. We'll also tackle forest plots, which are the visual summary of multiple trials combined. By the end, you'll know how to read a forest plot, spot bias risks in trial design, and ask the right questions before recommending a drug to a client. Here's what we're covering today. Study design is the skeleton of any clinical trial. If the design is flawed, even beautiful statistics can't save it. We'll look at what makes an RCT trustworthy, what can go wrong, and then we'll get hands-on with forest plots. Let's start with a question every clinician should ask before reading the results section. Can I trust how this study was done? Randomization matters with a simple example. Imagine you study pain medication in dogs, and you let clinicians decide which patients get the drug. Well-meaning clinicians might give the drug to sicker patients. Now your drug looks worse on outcomes, not because the drug failed, but because they started sicker. That's selection bias. Randomization fixes this by flipping a coin, metaphorically. The drug group and control group are built by chance, so on average, they look the same at baseline. Any differences in outcomes at the end of the study are then attributable to the treatment itself. It's the only design that lets us say the drug caused this outcome. Everything else can only show association. Walk through the key features of a good RCT. Think of this as your quality checklist when reading a drug trial. Randomization we've covered. Blinding is the next big one. If the owner knows their dog got the real drug, they may report better outcomes even if the drug didn't actually work. This is the placebo effect in veterinary medicine, and it's real. Pre-specified outcomes matter because if a researcher measures 20 outcomes and reports only the 3 that were significant, you'd expect 1 or 2 to be significant by chance. This is called p-hacking, and it inflates the false positive rate. Intention to treat analysis means you keep all randomized animals in the analysis even if they dropped out. If you only analyze completers, you introduce bias. The animals that tolerated the drug to the end may be systematically different than those that didn't. Your practical bias checklist. When you pick up a drug paper, flip to the methods section and run through these questions. Was the study blinded? If owners scored pain at home with no blinding, those scores may reflect expectation more than drug effect. Was there a control group? A study showing 70% improvement means nothing if 60% would have improved on their own with rest and supportive care. Look at Table 1 in any good RCT. It should show baseline characteristics. If the groups look very different at the start, something went wrong with randomization. And always check funding. An industry-sponsored study isn't automatically wrong, but a pattern of only positive results from a sponsor's own trials should prompt healthy skepticism. Forest plots appear whenever researchers pull multiple studies together. This is meta-analysis. The logic is one study might be wrong, but if multiple independent studies all show the same thing, we can be much more confident. Each row in a forest plot represents one study. The horizontal line is the confidence interval. The box in the middle is the point estimate, and its size is proportional to how much that study contributes to the overall answer, usually based on sample size and precision. The diamond at the bottom is the key result, the pooled estimate. Its width shows the CI for that pooled estimate. If the diamond doesn't cross the null line, the pooled result is statistically significant. Let's look at a real example. This forest plot for a hypothetical drug in canine lymphoma, reading left to right. All point estimates are below 1, meaning the drug reduces the odds of the event compared to control. That's in the right direction. Patel et al. Has the widest CI. Look at their sample size, only 60 dogs. Wide CI means uncertainty. Garcia et al. With 310 dogs has the narrowest CI and the largest box. The pooled diamond at the bottom, OR 0.63, confidence interval 0.52 to 0.74, the diamond doesn't cross 1. This is a statistically significant benefit. Now ask, are the studies consistent? If results were all over the place, some showing benefit, some showing harm, that's heterogeneity, and it complicates interpretation. Here, they look pretty consistent, which is reassuring. One more important concept before our case, heterogeneity. Even if 5 studies all test drug X versus placebo for pain, they may have different breeds, different doses, different pain scales, different follow-up periods. When studies give wildly different results, the pooled estimate can be misleading. The I-squared statistic quantifies this. Under 25%, studies are reasonably consistent. Above 75%, the studies are so different that pooling them may not make scientific sense, like averaging apples and oranges. Always look for this in a meta-analysis. If I-squared is high, read the individual studies and see which patient population most closely resembles your own patient before applying the results. The case question, take a moment to think through this. You're looking at a forest plot of 4 RCTs evaluating meloxicam for feline osteoarthritis. 3 studies show consistent benefit, odds ratios around 0.55 with CIs that don't cross 1. The 4th study has an OR of 0.44, even larger apparent benefit, but the CI is very wide from 0.15 to 1.3. It crosses 1. Why? Look at the sample size, only 28 cats, too small to be precise. This study is not contradicting the others, it's just underpowered. The point estimate actually agrees with the trend. The pooled OR is 0.54, meaning meloxicam approximately halves the odds of persistent pain compared to control. The CI is 0.41 to 0.68, doesn't cross 1, and I-squared is 18%, which is low. The studies are consistent. Interpretation. This is a coherent body of evidence supporting meloxicam for feline OA pain. The small outlier study doesn't undermine the conclusion, it just contributes less to the pooled estimate. Would you recommend it? With appropriate dosing, renal monitoring, and informed consent? Yes, this evidence supports use. The key lesson is this: don't let one underpowered study shake your confidence in a consistent pooled result. To wrap it up, RCTs are the most trustworthy tool we have for evaluating drug efficacy, but not all RCTs are equal. Blinding, randomization, pre-specified outcomes, and intention to treat analysis are your quality markers. Forest plots let you synthesize multiple RCTs into a single answer. Read each row, check the confidence interval, note the box size, and always look at the diamond and I-squared. In Talk 3, we move to survival curves, one of the most common graphs in veterinary oncology and critical care. See you then.